Hello guys, welcome to my YouTube channel. So today we are going to see colony immunoassay. So it is also called as immunological screening of a gene library. So it is basically similar to ELISA or Western blot assay. Okay. So last step of Western blot assay or the ELISA assay. So if you know Western blot or ELISA, then it is basically the same thing. So we start with step one. So I think all of you know bacterial transformation. So what happens in bacterial transformation? You let you let a plasmid get introduced into a bacterial cell, and if that bacterial cell takes up that plasmid, which is generally a recombinant DNA containing some antibiotic resistance gene within it. Okay, so if that bacteria takes up that recombinant DNA containing the antibiotic resistance gene, then that bacteria will be those bacterial cells will be able to grow on a selective medium of containing antibiotics, and those bacterial cells which will not take up that foreign plasmid into them, so those bacterial cells would not grow on that selective medium, so only the transformants will grow and the untransformed cells will die. So this is such kind of a plate. So this is a transformant plate where these colonies that you can see here, these 7 colonies, uh, these 7 colonies that they have shown, these 7 colonies are basically transformed colonies. So you transfer the cells to a matrix. Now this matrix is usually a nitrocellulose or a nylon matrix. Okay, so similar to blotting methods that you use, you gently press the matrix or the membrane over the colonies, and you wait for some time, you give some buffers and all these things, and the colonies get transferred from the media, from this agar media to the nylon matrix. And then the second step involves lysing the cells. So after the colonies get transferred to the matrix, you lyse the cells with some lysing agent which breaks the cell wall, and you let the proteins bind to the matrix. So there are various binding methods available, which utilizes which makes the proteins bind to the matrix strongly, and the unbound proteins are washed away. So only in the matrix what you see, these are the bound proteins which binds to the matrix. Now after you have the only bound proteins on the matrix, the third step is to treat the matrix with primary antibody. Now this antibody is specific to the protein, so this matrix, this antibody should be such an antibody which should bind to this bound protein already present within the matrix. So here, as you can see, these are the places where the primary antibodies uh, bind to the protein. Okay, now after giving the primary antibody, you wash once to remove all the non-specifically bound primary antibody. Okay, and then you give the secondary antibody. Okay, so what will happen, the secondary antibody, so these are the secondary antibodies E, which have been bound to the primary antibody C. The first, the primary antibody binds to the protein, and the secondary antibody binds to the primary antibody. So these are the places. See, there are 2 antibodies. Okay, so then you again perform washing step to remove all the non-specific bindings. So there may be other secondary antibodies which did not bind but are present, so you remove them, and then you give a substrate. Okay, so substrate means if the secondary antibody binds to the primary antibody, then it should convert one substrate into another form so that you can get some fluorescence or color. Okay, maybe a fluorescence or a color within the reaction medium. So if you are getting this color or a fluorescence, that means that that particular protein is present within that medium. Okay, so this is a confirmatory step. So that protein is present. Because of that, the primary antibody has been bound to the protein, and because of the primary antibody, the secondary antibody has been bound to the primary antibody, and it is showing color because it has converted some substrate which is already given in the medium. So this is an indirect method of a confirmatory test that your protein is present, your desired protein is present, and then you can go on for subculturing and you can preserve them, or you can perform some experiment if you wish to. Okay. Cells from this positive colony can be subcultured because they may carry the plasmid insert DNA construct that encodes the protein